Pleased to introduce Michael Golway. He's the president and CEO of Advanced Solutions Life Sciences. He's a talented entrepreneur, engineer, and inventor. He has led 10 acquisitions and as well as started 6 startups. He's the lead inventor of the BioAssemblyBot and the 6-axis robotic 3D printing system. If any of you guys have been to ARMI on site, you've probably seen that robotic system upstairs of the main offices. He'll be talking about a project that was funded under an ARMI award entitled a Generalized Biomanufacturing Platform for Dynamic Vascularized Tissues. Please welcome Michael Golway.
Thank you, Mary Claire. Good afternoon, everyone. I am presenting from the beautiful city of Louisville, Kentucky today. And my presentation is on a subset of the project that Mary Claire just mentioned involving an artificial intelligence application. So I am going to take you on the journey through this discussion today, really through 3 parts. I'd like to share with you some background information, talk to you about the solution, and then provide a use case. So let's start with the background. The work product shared today, as mentioned, is a subset of a project that is actually in process, this generalized biomanufacturing platform for dynamic vascularized tissues. So the due date for this project is next January, January 30th, 2021. I do want to take just a minute to thank our partners on this project. There's an enormous amount of work that has already been put into this project, including the entire ARMI team that we interface with. It's such an awesome group of professionals. In particular, on the leadership side, the support from Tom Bolenbach, Maureen Toohey, and Dean Kamen. From my team on the project I'm going to share with you today, I'd like to thank Rob Eli, who is a partner and executive VP for artificial intelligence and intellectual property. The engineers that worked on this project, including Victor, Alex, Christian, and Marcel, and then Dr. Hoyne, who is our chief scientist. And then finally, I want to thank Paul Goodwin at Cytiva, which was formerly GE Life Sciences. Paul also serves on the ARMI Leadership Advisory Council, and he's been a valuable friend and partner to our team for this project. So the background information that is most important for the artificial intelligence work product involves really 3 components of the Agile biofabrication work cell. Number 1 is the BioAssemblyBot, which enables us to manufacture and make tissue with a 6-axis robot arm. The second piece is the Cytiva N-Cell, which is a high-speed confocal microscope that enables us to image the biology. And then finally, the BioStorageBot, which is a multi-compartment incubator that also allows us to simultaneously perfuse tissues. Now, it's important to understand the workflow automation that exists in this Agile manufacturing work cell and how the AI was built for this big data application to help our team understand and gain insight into the science, not only in an efficient manner, but in ways that allows us to materially advance the science. So let's take a look at a video so you can see these technologies working together. Okay, so what you see here is, first of all, the Advanced Solutions Bio apps. This is a Visio flowchart drag and drop event processor. We like to think about the workflow, the automation workflow as a recipe. And so all the steps involved in building the recipe, there's a really easy drag and drop. And we're actually switching back and forth between the bio apps and the actual physical biomanufacturing work cell, or biofabrication work cell. And so we see a series of steps where the robot is performing activities within the work cell. Here you see the robot has handed off a 384 well plate to the N-Cell analyzer, and N-Cell analyzer is able to quickly process the specific area and the biology of interest, and then we get that data, and that is really a feed point for our AI that we're going to discuss today. But in conclusion here, the robot also has the ability to take our biology and move it back to our biostorage bot into a specific compartment for incubation and perfusion. So the final piece of background information involves Advanced Solutions angiomics microvessel technology, which was pioneered by Dr. Jay Hoyne all the way back in 1986. He discovered and patented a process where microvessel isolates are harvested from adipose tissue. And this is typically belly fat that we harvest from. And then we use that to recapitulate native angiogenesis and vascularization. And we've done that both in vitro and in vivo. And the angiogenesis part is obviously where we get brand new blood vessels to grow. And so he's dedicated much of his professional career to the advancement of vascular biology and angiomics. And as a result of this, he has over 50 refereed articles. In addition to that, we also have customers around the world that have published in over a dozen refereed articles related to this technology. So in over 2 decades, the angiomics vascularization research has been applied to tissues and cells from nearly every organ in the human body, including applications involving things like beta insulin producing cells. You can see here liver cells, even tumor cells. And we found the microvessel isolates from belly fat are actually very plastic and adaptive when we place them in different tissue cell systems. So as an example, if we mix our angiomic microvessels with hepatocytes, in about 14 days, we get a vasculature that is liver-like. Now, on the right hand of the screen, you can see that when we mix our angiomic microvessels with astrocytes, which are brain cells, in a couple weeks, we get vasculature that has blood-brain barrier characteristics. So therefore, the angiomics vessels are a raw material for the ARMI grant with our Generalized biomanufacturing platform for the vascularized tissues. Okay, so this leads us to the artificial intelligence problem we are working to solve. There's a large engineering and scientific effort behind the BioFab workflow, the automation and systems integration for the ARMI project. The balance of this presentation is dedicated to the tissue cell assessment. This is, and that is really how do we improve our ability to assess the biology from the microscopic imaging. So let's dive into the solution piece of this. We built our AI solution around the following design criteria. Number one was throughput, and that is how many measurement samples can be processed in a specified time period, including the prep time for the samples. Number two is the analysis. That is the time required to analyze the imaging. And three is the accuracy, and this is simply the measured value minus the expected value over the expected value. Number four is our ability to analyze images in 3D. And the fifth design criteria was the flexibility to adjust to different tissue and cell constructs. Another design criteria was to bring forward an ability to consistently and quickly measure tissue cell characteristics over time. So here you have a plot of the different tissue characteristics, which can include things like quantifiable measurements like number of cells or the length of cells, and being able to measure those samples over time. So for the purpose of this project, there were really three alternatives that we considered. One is the human, in which we use a microscope to visually assess the desired tissue characteristics. The second was pixel software. Here we're taking a gray level image of the biology. We're dividing it up into a pixel grid, and the grid is really determinant of the resolution. And with each pixel, there's a gray level value from 0 to 256. And from that, software algorithms are written to assess each pixel and determine the desired tissue characteristics. And then finally, our artificial intelligence and machine learning alternatives. So with a machine learning application, this is where we are acquiring data from a machine. So in this particular project, the machine that we're acquiring the data from is the Cytiva in-cell imager. And then the artificial intelligence is the model that we're teaching to make decisions based on that data that we achieve. So when you compare the three alternatives relative to the design criteria, you can start to see that there's some non-desirable constraints that are now not well suited for our agile biofabrication work cell. So the human, as an example, is fairly accurate, has high flexibility to assess different biology structures. However, there are constraints on things like throughput, analysis time, and even 3D capability. The Pixel software is constrained from a throughput standpoint because you still need a human to build the samples. And the analysis time is a little faster, but the accuracy is significantly less than a human. It has a moderate ability to incorporate 3D analysis, but the flexibility is very low because with each tissue and cell scenario, it has to be programmed with an algorithm. The artificial intelligence and machine learning segmentation approach really has three parts to the solution. So the first is the desktop application. This is where the user interfaces with the machine learning and trains the artificial intelligence model. The training occurs through a series of annotations in which an image is presented, and then the user simply identifies the biology of interest. In this particular example that you're seeing, we are teaching the AI to identify microvessels. So the training of the AI occurs over a number of human annotations and image sets. The second step occurs in the cloud, where the AI learns from the human. And we're using Microsoft's Azure AI platform. Now, once the AI model has been trained, it can be loaded back on the desktop, and then you can feed it new images and really assess how well did it learn, and you can assess its accuracy. So through this iterative process, the AI grows increasingly intelligent and can begin to match, and in some cases, surpass the accuracy of a human. Now, once we have the AI trained, we can integrate it into our agile generalized biomanufacturing platform and now begin to use the decisions that the AI makes to control even the robotic arm and the automation workflow. So based on the video we viewed earlier, you may start to see that we have incredible power to not only quickly assess the biology, but we also can assess the biology over time with our build capability, our image capability, and our closed loop incubate capability. All controlled, all the material flow is controlled through our robotic arm. Okay, so let's move on to the use case. Our angiomic vessels are very dynamic, and consequently, they need to be assessed over time to determine the viability. So we come back to our chart here. In this case, our tissue characteristics are things like new vessel, vessel length, and quantity. And so in this next video, hopefully you'll begin to appreciate the value of assessing this biology over time. Okay, so what you're looking at here is a series of images that were filmed over a 6-day period and stitched together in approximately 2.5-hour increments. In the beginning, you can see individual angiomic microvessel isolates called the parent vessels in red. Over time, you see the parent vessels begin to sprout and undergo angiogenesis, or growing to new blood vessels. And then by the end of the 6-day period, the biology structure and the state is much different than it was at time zero. So understanding these tissue characteristics over time, again, number of new vessels, the vessel length, overall quantity of the vessels is critical to building the vascularized tissue. And each of these characteristics were taught to our AI. Now, within the AI model, we can also teach it new characteristics. So in a broad sense, we're teaching it about microvessels, but we can also teach it to also identify things like new microvessels. So that same 3-step process is employed with step 1 in that image on your left-hand side, which is creating the polyline annotations of new vessels, and then the AI beginning to learn how to identify these structures through the object annotation and detection step. And then once the trained model is in use, it's able to identify the new characteristics with polylines. Now, this provides for accurate count and length, obviously, but the output from the AI also provides the user with a visualization check as to the accuracy, and frankly, it gives a lot of confidence to the user that the model is sufficiently trained. I will also share with you that we tested our AI segmentation across different imaging modalities. In this slide, you see 3 images. The first was obtained from the in-cell scanner. This has a 10x magnification. What's really amazing about this is with only 79 images, the AI began to identify vessel lengths with a 90% accuracy. The second image you see is from a phase microscope that has a 10x magnification. You can see that with 60 images, the accuracy was 84%. Certainly the grayer background here impacts the image accuracy, which you have to accommodate with additional cycles, additional repetitions, so that the AI can learn to segment the less clarity that you see with that first image. Distinction between the background, I should say. And then the third image is a confocal microscope, also a confocal microscope, but notice that it's at a 4x magnification. That also impacts the accuracy. So we know that with the higher magnification, we get better accuracy. It's easier for the, and that's intuitive. That also applies with humans as well. But it's important to note here that only with 54 images, the training was already yielding a 75% accuracy. Okay, so a lot of data on this slide. I just want to show and share with you that this is one example of many examples where we have used design of experiments in our own labs that we've completed to assess the accuracy of the AI segmentation. So in this one example, just wanted to give you a flavor for how we do this. In this particular example, our team is growing microvessels in 4 experimental groups. And what we're really trying to do is attempt to understand vessel growth through a tissue interface. So you might think of, if we need to grow microvessel or blood vessels through an organ wall, as an example, how do we best do that? And so this is a design of experiments that helps us understand that. And in this case, we're using stromal cells. So the tissue characteristic that's important to measure for this experiment is vessel length. And what you see here are 3 particular measurements. One is the black bar, which is the result of counting the vessel length by hand. The gray bar is the results obtained from the AI, and the white bar is obtained from the Pixel software. So in this example, you can see the AI had an average accuracy of about 93% with small variance. The Pixel software had an average accuracy of about 50%, and there was quite a bit of actually significant variance. Now, we're in the early stages of teaching our AI model or our AI platform to also perform 3D analysis, but you can see in this next video that not only are we able to identify vessel structure in 2D, but we're beginning to make the connection to 3D, and the AI is drawing its decisions in a solid 3D model. So this is an AI's decision and interpretation of the data. So we're especially excited about this capability because we will not only be able to increase tissue characteristic accuracy for things like vessel count and vessel lengths in which they go across different planes, in a true 3D space, but we'll also be able to calculate vessel volume And actually, we believe eventually we'll be able to measure vessel lumen size, which is the opening inside the blood vessel. Now, all these measurements give us the ability to significantly advance our ability to understand vessel behavior in a variety of tissue environments and gain an understanding of vessel cellular biology to better predict outcomes for a variety of tissue designs. Now, one of the best indicators of a new tool effectiveness is to put it in the hands of a professional. So the next video here, I want you to hear it directly from Dr. Hannah Strobel, who is a postdoc fellow working at Advanced Solutions in our Manchester, New Hampshire lab. And she's going to share with you her direct experience with the AI segmentation software.
Measuring the length and density of microvessels can be very challenging to do in an automated fashion. Sometimes it is difficult to distinguish between a microvessel and an elongated cell or a small piece of tissue. Because of this, I was initially a little skeptical of this software. We have tried other methods, other softwares in the past to measure vessel density that have been unsuccessful. But with the vascular segmentation program, it actually learns what a vessel looks like, much like a person can. Once it's trained, it can analyze an image in seconds. And since I often have 50 to 100 images in a data set, this saves me hours, if not days of time with each experiment. After using and validating it on my own data sets, I've found that it's accurate enough that it will even pick up on vessels I may have missed in my own annotations. And this has made me confident and happy that I now have an automated means to rapidly and accurately quantify vessel density in my cultures.
I really appreciate Dr. Strobel's assistance with this. It's invaluable for our engineering team as we went through the development process. So in conclusion, we are very pleased with the AI segmentation tool as an enhancement to our general biomanufacturing platform. For the dynamic vascularized tissues. There's still a lot of work to do, but we gain high throughput capability by integrating the AI segmentation with our BioAssemblyBot 6-axis robot, the in-cell imaging, and the BioStorageBot incubator and perfusion platform. You heard from Hannah that once the AI was trained, the analysis time was short. Through our experimental designs, we've been able to show an accuracy at, or in some cases above, a human accuracy. And then the 3D analysis, although it's early in development, it's already showing some strong potential to provide a high level of capability compared to human and pixel software. And then finally, the AI segmentation is highly flexible in its ability to learn new biological structures. So we showed you today an application for blood vessels, but it can be trained to learn many types of biological structures. It does not, I should say it does need to be trained like a human, but the AI over time gets better and better, never regressing and only continually improving towards a mastery level. So thank you for your time and attention today. I did put my email address up there if you'd like to reach out to me directly for any questions that are not addressed in our Q&A. And with that, I'll turn it back over to Manchester. Thank you.